Welcome to Biocomputation with Dr. Max. I want to share an important story related to cancer treatment in the USA. A friend of mine asked me to provide technical support to their friend who was diagnosed with triple-negative breast cancer. Triple-negative means that none of the three hormones which can promote cell growth and neoplasms are the driving factor of this cancer. Before starting to provide a review of medical information, I pointed out that the majority of precision medicine is genetic, and most mutations no known significance are known, in fact, as missense mutations and may be silent and not affecting the activity of the protein product. To my surprise, there was a critical mutation in the p53 protein, the so-called guardian of the genome, also known as the master tumor suppressor. The surprise was twofold. First, finding a strongly actionable mutation is rare. Second, finding a mutation that is not already properly annotated and acted upon with best clinical practice is even more rare. Yet such a mutation and total misinterpretation of its significance was staring me in the face. A precision oncology company based near Chicago is focused, called Tempus, is focused on interpreting genetic information. Now they're called Tempus AI, of course. The mutation in question is R248W, and it is recorded as LOF, meaning loss of function of the p53 protein with mutation of arginine at position 248 to tryptophan. This interpretation loss of function is surprising in light of abundant scientific evidence that suggests that it is actually gain of function. This difference is not semantic. The mutant has novel functions that actively contribute to tumor progress. More importantly, after chemotherapy and surgery, the proposed method to treat the residual disease is radiation therapy. This is completely wrong for this individual. Despite a mountain of evidence, which I summarized in a PDF and will go over in a separate video, which will be long and heavy on science, oncologists and radiation oncologists on the case are sticking with standard of care, namely radiation. I also consulted a friend of mine who is a cancer doctor specializing in breast oncology with interest in radiation therapy, and he essentially told me the same thing. So let me explain why this gap is important. p53 is mutated in roughly half of all human cancers and has over 80% prevalence in triple-negative breast cancer. R248 is the most frequent residue to be mutated. It is the hottest spot of arguably the most important oncogene. There is one test of radiation resistance which was recently approved by the FDA, but as often happens, it's not precisely suitable. And that's in two ways. It's called Decision RT, and it's a 7-gene biosignature test designed to predict the risk of cancer recurrence and the benefit of radiation therapy for patients with ductal carcinoma in situ, DCIS, following breast cancer conserving surgery. So it would be appropriate, except for the fact that the patient in this question has been diagnosed with invasive ductal carcinoma, not in situ, meaning in place. So DCIS does not apply. And indeed, p53 mutations result in more invasive cancer types. So this description, which is an assessment of histological assessment from analysis of tissue slices, could have been predicted from genomics, namely invasiveness. There is another near suitable test called Oncotype DX. It is not FDA approved, but operates as a laboratory developed test. I've actually been in charge of tests like that for cancer. They can be quite effective, but they have limitations, especially in terms of the breadth of application. There is a centralized credentialed laboratory that can run these tests, and they can keep some aspects of it as essentially a trade secret, although it has documented results. While this test, Oncotype DX, is best known for guiding chemotherapy decisions, studies also show that low recurrence scores do not see a statistical survival benefit from radiation, whereas those with intermediate or high scores do. Another limitation is this test is primarily applicable to early stage estrogen receptor positive, ER positive, and HER2 negative invasive breast cancer. We're talking about triple negative breast cancer. So it's negative not only for HER2, but also for estrogen and progesterone. These examples show how highly specific tests leave many patients without coverage by developed diagnostics. Faced with this uncertainty and potential liabilities, clinicians resort to standard of care therapy. Let me summarize. Radiation therapy is anathema to a person with R248W mutation. And again, I go into that detail more deeply, but I will cover it briefly here in just a minute. Unfortunately, this mutation is an aggressive driver of tumor progression. There are dozens of therapies, specifically small molecule therapies, that could target mutant p53 to either restore its native function or at least clear it. That could have a good chance of helping the individual. This interpretation as loss of function completely misses the major therapeutic opportunity to target mutant p53. Radiation therapy would only result in acceleration of Darwinian evolution of cancer cells, shortening the lifespan of the patient. So let me present very briefly the summary of a scientific argument and thereby a challenge to the scientific establishment. Let me speak briefly and plainly. Ionizing radiation kills living systems. It is one of the primary limitations of humans living in space. We also know this from unfortunate results in Hiroshima and Chernobyl, which showcased not only lethality, but long-term exposure problems resulting in cancer, specifically for survivors. This is the reality in the nuclear age, that if the radiation doesn't kill you, it could cause cancer. Of course, in both of those cases, we're talking about uncontrolled exposure, location on the body, and dose. Therapeutic radiation is able to provide some health benefits because biology is exposed to it is not just an object, but is the subject of radiation. The body has to actively respond to preferentially eliminate cancer cells in response to DNA damage, and the dose must be manageable for cells repair machinery for healthy cells to avoid developing cancer. To summarize, radiation therapy heals the body only if the body is able to actively respond by selectively eliminating cancer cells. R248 p53 mutant through several well-established mechanisms, and the body is unable to respond properly. Putting this situation into larger context, we can see that there's a chicken and egg dilemma where more personalized cancer treatments are not manifesting, and we can break this dilemma through data sharing. The positive and the negative experiences of patients with various cancer types with, for example, this mutation, especially in the context of triple negative breast cancer, where it is highly prevalent, could accumulate clinical data to sway both doctors and regulators. As things stand, this mutation and its impact has been known for at least 2 decades to be gain of function, not loss of function, with important clinical implications, yet this information has not percolated into clinical practice. It appears that until regulators approve some tests, there is no recourse for patients with these mutations. Diagnostic tests such as DecisionRT and Oncotype DX, or potential complementary diagnostic tied to specific drugs, are highly specialized and do not support all patient types. Thus, it is up to the patients themselves to participate in platforms that can share clinical-grade medical data while preserving their privacy. The right type of innovation, which empowers patients while enabling sharing and monetization, will not come from big business or big government. It is a ground-up effort to endow the patients with tools for management and ownership of their medical data. Please like, share, and subscribe.